Chapter 9 is on carbohydrates, which is a challenging topic, but one of my favorites. As I usually do, I would first mention that there are several functions of carbohydrates. First, they are an energy source for both plants and animals. One of the first pathways we will examine is glycolysis. They are used as a source of carbon in metabolic processes. For example, sugar is converted into fat. Third, they are a storage form of energy in both plants and animals, though the molecules used differs. Fourthly, they are structural elements of cells and tissues, as we will see later in this chapter when we refer to the glycocalyx. And finally, they play a role in recognition, particularly in the immune system. Carbohydrates were originally thought to have the formula CH2O, but that definition did not work well for very long. Only the simple monosaccharides obey this rule. Therefore, the definition that I use is that a carbohydrate is a polyhydroxyaldehyde or ketone, or a larger molecule that can be hydrolyzed to a polyhydroxyaldehyde or ketone. By this definition, the simplest carbohydrates must have 3 carbons, and the 2 simplest carbohydrates are shown on this slide. On the left is what we would call an aldolase because it contains an aldehyde group, and on the right we would have a ketone, which contains a ketose, which contains a ketone group. Both of these would be referred to as a triose because they contain 3 carbons, and they're carbohydrates. Therefore, a complete name for the left molecule is an aldotriose, and the right molecule is a ketotriose. The names of these 2 particular monosaccharides are called glyceraldehyde and dihydroxyacetone. It will be noted that the glyceraldehyde has a chiral carbon in it, and therefore can exist as 2 different stereoisomer forms, shown on this slide. In biochemistry, we use the DL system, which is an older system than the RS system used in organic chemistry. The beauty of using the DL system is that when the OH, that group that is on the central carbon, is on the left-hand side, we refer to it as an L sugar. L is short for levorotatory. When the group is on the right, is referred to as a D or dextrorotatory group. And I and many other biochemists will refer to these as right and left-handed sugars. If we start looking at more complicated molecules, we would run into erythrose and threose as the 2 aldotetroses. Erythrose can readily be identified as a D sugar, but if we look at threose, we might be confused about whether it is a D or an L, as one of the OHs is on the left-hand side. Therefore, remember that we always look at the OH group furthest from the carbonyl group when determining whether a sugar is D or L, and both of these are D sugars. This is an opportune time to note that nature uses almost exclusively D sugars. There are a few other terms used in stereochemistry which we should be review, and they're not used heavily, but they must be touched on. Enantiomers are mirror image molecules, and very importantly, a D and L sugar are enantiomers of each other. Another term is diastereomers, which are non-superimposable, non-mirror images, and I include that definition only for completeness. Epimers are a very important concept in carbohydrate chemistry, and they differ in arrangement around one other chirocarbon. I will point out epimers as I proceed through this lecture. The molecules that I have shown you thus far are presented in a Fischer projection. These are named after the infamous German carbohydrate chemist Emil Fischer, who established the convention that vertical bonds are pointing away from you towards the back of the screen, and horizontal lines are pointing out towards you. Remember this as your loving biochemistry teacher standing in front of you and reaching out to give you a hug. Carbon number 1 is that which has the highest priority functional group, which is the aldehyde group. This structure is not one that you should commit to memory. The reason for this is that most 5 and 6 carbon sugars, which includes most sugars in biological systems, are in a ring form and most commonly in the chair conformation. These molecules will be referred to as pyranoses and furanoses because of their similarity to the molecule pyran and furan. An extremely valuable skill is to be able to convert Fischer and Haworth projections. Haworth projections are those showing the sugar in the ring conformation. The ring forms because the carbon of the aldehyde group reacts with the lone pair of electrons on the carbon 5 OH group. The hydrogen of the OH group is transferred to the carbonyl oxygen to form a new OH group. The carbonyl oxygen becomes part of the ring, and because the carbonyl can be pointing up or down when the ring closes, it is possible to form alpha and beta anomers. We can very methodically step through the way a ring forms, but it is useful to know a shortcut. And that is, if we see a 6-carbon aldose, such as the one shown on this slide, we should automatically think it will occur in the pyranose form. Secondly, in D sugars, the number 6 carbon is always pointing up. Then we think about the expression right down and left up. In this case, the number 2 and 3 OH groups are pointing to the left, so they will be pointing up into the ring, and the number 4 carbon is to the right, so it will point down. The number 1 OH group can be either up or down, and if we do not wish to specify, then what we do is have the OH group pointing straight out as shown on this slide. Sugars have a preference for existing in either the alpha or beta anomeric position. An alpha anomer has the two groups on the 1 and 6 carbons pointing in the opposite directions. Beta anomers are pointing in the same direction. I keep this straight by thinking A and O are both vowels and B and S are both consonants. The ring structure can open back up, and therefore is in equilibrium between the open chain form, alpha, and beta structures. In the case of glucose, about 2/3 is present in the beta conformation, and 1/3 is in the alpha conformation, with less than 1% existing in the open chain form. The percentage in each of these forms can be determined experimentally, though it is not the easiest technique in the world. To give an example of how this works, if you have a sample of pure beta-D-galactose, it will have a specific rotation of 52.8 degrees. A sample of alpha-D-glucose has a specific rotation of plus 150.7 degrees. However, in solution, these will be in equilibrium with each other, as previously pointed out. After this equilibrium is reached, the observed specific rotation will be plus 80.2 degrees. This is referred to as mutarotation because of the change in specific rotation. I would note to you that because the rotation is 80 degrees, there should be more beta than alpha because 80 is closer to 50 than 150. Through some algebraic manipulation, it can be determined that there is 28% alpha and 72% beta. I would not have you calculate this on an exam. Next, we will introduce the important simple monosaccharides, the first of which is glucose. I am showing glucose in the form that is most physiologically important, and I would have you learn this structure, which is alpha-D-glucose. The second important monosaccharide is mannose. This is an epimer of glucose in the number 2 position. You will notice that it points up into the ring instead of down like glucose did. Third, galactose, which is an epimer of glucose at the number 4 position. And this is also an alpha sugar. Fourth is fructose. Fructose is a ketohexose, and ketohexoses are furanoses, most often anyway. I would also point out to you that this is the beta form of fructose, since the right-hand OH group, which happens to be the number 2 carbon in this case, and the number 6 carbon on the left are both pointing upwards. The fifth and last structure that I would have you memorize is that of beta-D-ribose. The reason for memorizing these 5 sugars is that they are the basis for many other important monosaccharide derivatives. For example, deoxyribose is the same as ribose, except at the number 2 carbon. For another example, which is rare but important, is L-fucose. Do not memorize the structure, but notice that the number 6 carbon is pointing down, which makes it an L, and is a CH3 instead of a CH2OH. It should be also noted that this is an alpha sugar. If you understand that this one is alpha, then you understand the alpha-beta system. There are also amino sugars. Glucosamine, which I am showing in the most important beta D form, galactosamine, which is not shown, N-acetylglucosamine, which is not shown, and N-acetylgalactosamine. You should convince yourself that you know how to draw the structure of galactosamine and N-acetylglucosamine from the information presented here. There are also oxidized sugars which are important. A lactone is when the carbon 1 oxygen is present as a ketone. A gluconic acid is when the C1 is present as a carboxylic acid, which can only happen in the open chain form. And glucuronic acid or glucuronates, which are oxidized to a carboxylate position at the number 6, C6 position. Note that the pK values of these are such that they are charged at physiological pH, and that's why we say gluconate and glucuronate to indicate that charge. There are other more complicated sugars which you should not need to commit to memory. In this particular case, I have N-acetylneuramic acid, which is one of a family of sugars referred to as a sialic acid. Monosaccharides undergo a number of reactions that are important physiologically. First, I will point out that monosaccharides are acid stable, but they undergo a process in basic solution which is referred to as epimerization. In other words, these are epimers that will form. If we begin with glucose, we will end with glucose, galactose, mannose, as well as some other unnamed sugars. As previously mentioned, they undergo mutarotation. Second, they can be oxidized to carbon dioxide and water, as known from high school biology. If a sugar has an aldehyde group, it is referred to as a reducing sugar. In other words, they will reduce substances such as copper 2 ion to metallic copper. Aldehyde groups can be reduced to an alcohol, which is referred to as a polyol. Polyol accumulation in diabetes contributes to diabetic complications. Alcohols will react with carboxylic acids to form esters, and I am showing here an example of a phosphoester. Finally, alcohols can react with other alcohols to form acetals. For this reaction, I am showing alpha-D-glucose in its chair conformation. I am showing the product as beta-D-galactose not because that actually happens in the reaction, but just so that you see what the alpha and beta look like at least one time. When the alcohol is another monosaccharide, then disaccharides are the products.